Welcome back to the deep dive. Today we are focusing on, well, what you could call the central nervous system's master regulator, the NMDA receptor.
Or NMDA, short.
Right, the NMDA. This channel isn't just fundamental, it's really at the crossroads of our highest cognitive functions. It drives things like learning and memory.
But there's a dark side to it, too. Flip that coin, and it's deeply implicated in the pathology of, devastating conditions like stroke and epilepsy.
So for anyone listening who's heading into neuroscience, pharmacology, or medicine, it sounds like this is a must-know topic.
It is. You absolutely have to understand how to measure, manipulate, and ultimately target the NMDAR.
Yeah.
The methods we use they're the keys to the kingdom.
Okay, so let's start with the basics. Structurally, what are we dealing with?
We're talking about ligand-gated cation channels.
Yep.
And they need two things to open up: the neurotransmitter glutamate and a coagonist glycine.
And they form these heteromeric complexes?
Yes, exactly. They always need that core NR1 subunit, and then it combines with one of the regulatory NR2 subunits A, B, C, or D.
And what's their defining feature? The thing that makes them so powerful?
It's their incredibly high permeability to calcium. That influx of calcium is what dictates everything, from strengthening a synapse to, unfortunately, triggering cell death.
So our mission today isn't just to list facts. We want to provide you with a practical toolkit, a manual.
Right.
We're going to synthesize the state-of-the-art molecular techniques, the logic behind them, the lab hacks, so you can walk away feeling comprehensively informed whether you're prepping for an exam or a lab meeting.
Let's get into it.
To start this journey, we have to begin at the beginning. How do you even get a functional NMDAR to study in the first place?
Right. And that brings us to the expression systems. The first, and really the pioneer, is the Xenopus oocyte, the frog egg.
The frog egg, okay. Why start there?
Well, the Xenopus oocyte system was a revolution in the 80s and 90s. Its main advantage is just sheer efficiency.
Efficiency in what way?
You inject the RNA for the receptor, and the oocyte is basically a giant, super efficient protein factory. It translates that RNA into functional protein in the membrane incredibly well.
And I remember reading that its size is a big deal here, too.
Oh, absolutely. The oocyte is enormous for a single cell, about 1 millimeter in diameter. And that's key for the classic method we use here, the two-electrode voltage clamp, or TEVC.
Because you need to physically stick 2 electrodes into it, right?
Exactly. One to monitor voltage, one to pass current. Its size makes that a relatively simple procedure.
Now, here's where the biochemistry gets really clever. I think this is the aha moment from back in the day. The oocyte has this built-in signal amplifier.
That's right. Researchers realized the oocyte was helping them out. When an express receptor gets activated, it can trigger the oocyte's own internal machinery.
The phosphatidylinositol pathway.
Yep, the PI turnover. And that, in turn, activates the oocyte's native calcium-sensitive chloride channels.
So wait, you're looking for a tiny current from your brand new NMDAR, but what you actually measure is this massive, easily detectable inward current from the oocyte's own channels.
Precisely. It turns a tiny little whisper of a signal into a loud, clear shout. It was an invaluable tool for cloning dozens of receptors that were otherwise really hard to detect.
It's amazing. So for anyone thinking practically, what's involved in using them?
Well, you have to keep the female Xenopus frogs happy. Tanks at 18 to 22 degrees Celsius, a strict 12-hour light-dark cycle.
And getting the eggs ready.
That's half the battle. You treat the ovarian tissue with collagenase to get the mature stage V or VI zoocytes. But here's a critical detail, a classic student mistake. It's about the RNA buffer.
Go on.
If you're using a tris buffer, you must not use DEPC to treat your solution.
Why not? What's the problem there?
DEPC is used to kill off RNA chewing enzymes, but it chemically reacts with tris. That reaction not only stops the DDPC from working, it can actually degrade your precious RNA. You end up with zero expression. It's a crucial little piece of lab wisdom.
Okay, so let's move from amphibians to mammals. We have cell lines like HEK293 and CHO cells. If oocytes are so efficient, why make the switch?
The main reason is cleanliness, biophysically speaking.
Cleaner how?
Mammalian cells, especially HEK293s, have far fewer interfering endogenous ion channels. So for high-resolution patch clamp recordings, they give you a much cleaner canvas to work on, a better signal-to-noise ratio.
But expressing NMDRs in mammalian cells comes with a huge problem, doesn't it? Excitotoxicity.
A massive problem. That big calcium influx we talked about will literally kill your transfected cells. They just die off.
So how do you protect them?
You have to bathe them in an antagonist.
Yeah.
The practical workhorse, especially for large cultures, is 1 millimolar ketamine.
Ketamine again. So why that over something more, I don't know, pharmacologically pure like APV?
Honestly, it's mostly economics and convenience.
Really?
Yeah. Ketamine is cheap, it's easy to buy in bulk, it's readily soluble, and it works really well. It protects the cells when expression is highest, and then you just wash it out before your experiment. Cell survival is the priority.
And just to round out the platforms, what if you don't want to do electrophysiology, but you need, say, big batches of pure protein?
Then you'd probably turn to the baculovirus system, using insect cells like SF9. That's what you use when you need milligrams of pure protein for something like X-ray crystallography.
All right, so once we have the protein expressed, we need to quantify its function. Let's start with electrophysiology. For our listeners, what's the key difference between whole cell and single channel analysis?
It's really the difference between looking at a whole crowd versus interviewing one individual.
I like that analogy.
Whole cell recording measures the macroscopic current, the total flow through every active channel on the cell. It's the population's average response.
And single channel.
That's the interview. You're documenting the molecular behavior, the actual gating, the mean open time, the flicker of a single protein molecule. It's incredibly detailed.
And the data from whole cell recordings, the dose-response curve, tells you a lot about how a drug works.
It tells you everything. You have your Emax, the maximum effect, and your EC50, how much drug you need for half the effect.
So if you add a competitive antagonist, I always picture it as two people fighting for the same chair.
That's a perfect analogy. Because they're competing for the same binding site, you can overcome the block by just adding more agonist, more glutamate.
So the curve just shifts to the right.
Exactly. Your EC50 increases, but the Emax, the maximum response, stays the same. You can still get there, it just takes more effort.
Okay, but what about an uncompetitive antagonist, like the famous MK801?
That's totally different. An uncompetitive blocker only binds when the channel is already open. Think of it like a cork you can only put in a bottle once the top is off.
So it traps the channel.
It traps it. And because it physically blocks the pore, it doesn't matter how much agonist you add, you can never reach that maximum theoretical current. So your Emax drops.
That makes it crystal clear. Now moving from current to, well, a quantitative shortcut. Ligand-binding assays, especially using radioactive MK801.
Yes, tritiated MK801. It's a hugely potent blocker that binds deep inside the channel core. And here's the crucial nuance. It can only get in when the channel is open.
So binding depends on the receptor being activated first.
Precisely. We call it a diva molecule. It won't show up unless glutamate and glycine have already rolled out the red carpet. This makes its binding super sensitive to the receptor's activation state.
Which means the assay conditions are everything.
Absolutely. If you do a short non-equilibrium assay, maybe 2 hours, you'll see that agonists increase the binding rate. But if you want the true equilibrium constant, the KD, you need a long saturation assay.
How long?
4 to 6 hours. And it has to be in the presence of saturating agonists to make sure you actually reach equilibrium.
And if you do that, what's the number we should remember for its affinity?
The equilibrium KD for MK801 is reliably around 1 nanomolar. It's incredibly potent.
And what's really fascinating is how sensitive this assay is to everything else. It's not just a glutamate-glycine switch.
Not at all. The channel is constantly being fine-tuned by its environment. The binding is sensitive to magnesium, to zinc, to polyamines, even to protons.
So pH matters.
A lot. The binding is highly sensitive to pH shifts, right around physiological pH, with a pKa of about 7.4. Tiny changes in acidity can dramatically alter receptor function.
Let's scale up. Drug discovery means screening thousands of compounds. This brings us to high-throughput screening, or HDS. The workhorse here is often the FLAPR system.
Right, the fluorescent imaging plate reader. It's all about measuring that crucial calcium flux we talked about.
How does it work?
You load your cells in 96 or 384 well plates with a calcium-sensitive dye, like fluo3. When you activate the NMDARs, calcium floods in, the dye lights up, and the machine reads the increase in fluorescence. It's a fast, scalable readout.
But there's a notorious practical problem here. Dye efflux. The cells just pump the guy right back out.
It's a huge headache. You can have a multi-million-dollar screening campaign fail because your signal just disappears over time.
So what's the solution? Is there a lab hack for this?
There is, and it's surprisingly simple. You add a carrier protein to your assay buffer.
Wait, really? Just protein?
Yep. About 1% bovine serum albumin, BSA, in the buffer. For some reason, it seems to inhibit that dye extrusion process. That tiny addition can be the difference between a successful HDS campaign and a complete failure.
Amazing. Okay, shifting from function to form. the actual physical structure. For years, the model for how glutamate receptors sit in the membrane was just wrong, wasn't it?
It was. Everyone assumed they had a 4-transmembrane domain structure, the 4TMD model, just like nicotinic receptors.
But the data didn't fit.
Exactly. The evidence, especially around the second transmembrane segment, the M2 domain, just didn't add up. It forced a complete rethink. We now know the M2 segment doesn't cross the membrane at all. It forms a re-entrant hairpin loop.
So it dips into the membrane and then comes right back out.
Like a needle and thread. And this led to the correct 3TMD model.
So how do you prove which parts are sticking out of the cell and which are inside?
You use tagging strategies. One is epitope mapping, where you insert a little tag, like a flag tag, that an antibody can recognize. If the antibody can bind without breaking the cell open, you know that part is on the outside.
And the other method is more chemical.
That's N-glycosylation tagging. You insert a sequence that tells the cell to attach a sugar molecule. Since that only happens on the outside of the cell or inside the ER, if you find a sugar attached, you know for sure that domain is extracellular.
Let's finish this section with how the cell itself regulates the receptor. Let's talk about calmodulin.
Calmodulin, or CAM, is the cell's essential calcium sensor. When calcium floods in through the NMDAR, CAM binds to 2 specific sites on the NR1 subunit's C-terminus.
And one of those sites is more important than the other.
The CBS1 site is the key. Binding there, which happens with an affinity of about 80 nanomolar, is directly responsible for what we call calcium-dependent inactivation.
And why is that inactivation so critical?
It's the brake pedal. After a big calcium signal, the kind that triggers synaptic plasticity, you need to reset the system quickly. CAM binding causes a 4-fold decrease in the channel's open probability. It stops runaway signaling and prevents toxicity.
So we've measured function and structure. Now let's talk about actively manipulating the receptor. Let's start with its sensitivity to the cellular redox state.
This is hugely important. The redox state, whether the environment is oxidizing or reducing, is mediated by specific cysteine residues on the receptor. And during something like a stroke, the redox environment changes dramatically.
So what's the chemical toolkit for studying this?
For the reducing side, our brake pedal is dithiothiol, or DTT. It keeps the cysteines in their reduced state.
And for oxidizing, the gas pedal.
That would be something like DTNB or nitric oxide donor like nitroglycerin. They oxidize those sulfhydryl groups and change how the channel behaves.
And how do you prove that the effect you're seeing is actually due to those specific sites?
You need a control. And for that, we use N-ethylmalamide, or NM.
What does MNM do?
It irreversibly latches onto those thiol groups and blocks them permanently. So you apply an NM first. If your DTT or DTMB then has no effect, you've proven that the change was mediated by those specific NMM-sensitive residues. It's the definitive control experiment.
Now for the really cutting-edge stuff, receptor engineering through biomimicry. As I understand it, scientists took a simpler receptor, GLER1, and started tweaking it piece by piece to make it behave like an NMDAR.
The goal was audacious, really. They wanted to see if they could transfer the unique properties of the NMDAR pore, its ion selectivity, its sensitivity to blockers onto the GLER1 scaffold. They focused on mutating key sites in that M2 reentrant loop.
And it actually worked.
Amazingly, yes.
Yeah.
They could functionally swap the pore properties between the 2 different receptor types.
And in doing so, they stumbled upon a huge structural insight, right? It solved a long-standing debate.
It did. By creating these chimeric subunits and seeing how they assembled, they could use this functional assay to figure out the subunit stoichiometry. How many pieces make up the whole receptor?
And the conclusion was
Monumental. It provided some of the strongest evidence that both GLUR-R1 and NMDRs are likely pentameric made of 5 subunits. It challenged the old 4-subunit tetramer model that had dominated the field for years. So engineering the function revealed the architecture.
What a journey. From translating mRNA in a frog egg to protecting HEK cells with ketamine, running binding assays with the diva molecule MK801, and using a BSA hack to enable high-throughput screening.
All the way to understanding calmodulin inactivation and re-engineering a receptor to discover it's a pentamer.
These methods really are the bedrock of CNS drug discovery.
They absolutely are. And to help reinforce some of this, I have a little review exercise for you at home.
Okay, let's hear it.
A new compound is tested on an NR1-NR2A expressing cell line. In a whole cell recording, it decreases the Emax, but doesn't change the EC50. At the same time, in an MK801 saturation assay, it decreases the affinity for MK801, so a higher KD, but leaves the Bmax, the number of sites, unchanged. What 2 distinct mechanisms might this compound have?
Okay, let's unpack that. The first clue is from the whole cell recording.
Right.
A drop in Emax with no change in EC50. That's the classic signature of an uncompetitive or non-competitive antagonist, something that's not competing with glutamate, but is reducing the total possible effect.
Good. That's mechanism one. Now, what about the binding data?
It makes it harder for MK801 to bind, it decreases the affinity,
Yeah.
but it doesn't reduce the number of receptors.
So what does that suggest?
It is just, it's not blocking the pore in the same way MK801 does. It must be an allosteric modulator. It's binding somewhere else, changing the shape of the pore, and making the MK801 site less
Yeah.
welcoming. So the answer is a compound that is simultaneously an uncompetitive blocker and a negative allosteric modulator of the MK801 binding site.
You got it. And that level of complexity is why you always need multiple assays.
Perfect.
And to leave you with a final thought, we talked about the receptor sensitivity to pH with that pK around 7.4. We know big changes in brain pH are bad, but what if subtle, localized shifts in pH, maybe around a very active synapse, are actually used physiologically to fine-tune synaptic function?
So not global regulation, but tiny microenvironmental tweaks.
Precisely. It encourages us to look beyond the big picture and see how the local environment might be a key to functional complexity.
A perfect place to end this dive. We encourage you to explore just how those microenvironmental changes could be the secret key to synaptic control.